Good afternoon, judges. My name is Dr. Shumik Biswas, and I'm the founder and CEO of Tenomix. As we all know, cancer is a terrible disease that personally affects each and every one of us in some way. In 2020, almost 27,000 Canadians were diagnosed with colorectal cancer. And shockingly, this ends up equaling to 73 Canadians being diagnosed every day. Now, when we look at the colon cancer staging process, part of it involves pathology staff manually searching through surgically removed colon cancer tissues for lymph nodes, which can take anywhere from 1 hour to over 2 hours for larger samples. Now imagine yourself in the shoes of pathology staff who are doing this tedious search process by hand, trying to find small lymph nodes that are the same size of a pencil tip. Does that sound reliable to you? Through customer discovery, we confirmed that this process is indeed tedious, expensive, and unreliable. And what's shocking is that lymph nodes can be missed in 30 to 60% of North American cases, which can lead to inaccurate cancer staging and improper treatment for patients. And pathology staff desperately need a solution that can help decrease the labor, reduce costs, and reliably locate lymph nodes. After hearing their pain points, at Tenomix, we've developed the Lymphinator, which is a novel benchtop robotic scanning device that automates the lymph node search process. With our patent pending technology, we're simply changing the tedious search process, which is the key bottleneck to accurate cancer staging. And by integrating early feedback from pathology staff, our device offers several benefits, which include seamless workflow integration, better lymph node detection, set and forget operation, and reduced costs for pathology labs. And this is how the Lymphinator works. Pathology staff only need to place a sample in our device, press the start button, and then our device will do the rest of the work, where ultrasound imaging and software are used to identify lymph nodes that then guide a robotic arm that marks lymph node locations for easy extraction by pathology staff. From our early experiments with our alpha prototype, we've been able to locate small lymph nodes that are difficult to find by hand. And the need for our device is also timely, because with increasing colon cancer cases, ongoing pressures faced by pathology labs, and the growing demand for lab automation, it's the perfect time for adoption. In terms of current competition, there are no direct automated competitors. However, there are alternative methods available, but they have significant drawbacks. By validating our device in the market, our device offers significant advantages compared to existing solutions, which include less labor intensity, cost savings, accurate lymph node identification, and increased throughput for pathology labs. Following a proven business model for medical devices, we'll use a B2B model involving sales to hospitals, which include hardware sales and a recurring revenue model with device consumables and software subscription. Based on comparable tech in the market and procurement thresholds, the sale price of our device is $100,000, and hospitals can see an attractive ROI of less than 1 year. For our go-to-market plan, since we've already engaged with several key opinion leaders, we'll first target large North American academic hospitals that have pathology teaching programs. These trainees will be trained with their device, and they will then be our advocates after their programs, which open the doors to non-academic and private pathology labs. The labs in Canada will be our initial validation market, and we're also actively pursuing several great leads in the US. Now, once expansion takes place into the US and global labs, we'd be looking at a massive $559 million opportunity each year. And I have to say that none of this work would be possible without my amazing team. With expertise in pathology, engineering, business, and medicine, my team and I have raised over $370,000 in non-dilutive funding, and we've already started clinical testing with human tissues, all within 8 months. We also have expert mentors in pathology, software, and business guiding us. Going forward, we'll continue with software development. And in terms of IP, we filed a provisional patent last July. So we'll convert this to a PCT this July to capture a larger market share by filing in countries with a high rate of colon cancer. From there, we'll then finish our beta prototype for a clinical pilot study early next year. And as we work with our consultants, we'll move towards a Class 2 regulatory approval and then plan for production and sales in 2024. Based on our 5-year financial projections, in the first 2 years, we'll focus on R&D activities and will require an initial investment of $1.2 million to move to regulatory approvals. Sales are expected to start by year 3, and we anticipate a 300% annual growth rate after that year. In terms of the Synapse Award, the funds will be used for prototype refinement, IP, software development, and for a clinical pilot study in Hamilton. This funding will help us reach 3 significant milestones, which you can see on the right. We also plan to use the in-kind services for legal support and further networking to grow the business. Having lost my aunt to improper colon cancer staging at a young age, this work is a tribute to her and millions of others who have lost their lives fighting cancer. My team and I are dedicated to help patients get the best quality cancer care. And here at Tenomix, we're ready to fight for cancer patients, one lymph node at a time. Thank you.
Great job, Samik. Thank you so much for that. You've given us lots of areas where we can ask questions, and what we'll do is we'll go around the judges one at a time and come back in a circle and see how many questions we can cover off. So maybe first of all, I'll hand over to Lou for the first question.
Thank you, Jane, and great presentation, Samik. Thanks. Just curious, you're starting with colon cancer. What other tumor types you think can be used for this device?
Lou, it's a fantastic question. So that's sort of the interesting application potential for our device, because for a lot of other cancer tissues, for example, head and neck cancer, breast cancer, lung cancer, gastric or stomach cancer, bladder cancer, they all require some form of lymph node dissection. So we definitely see our device being applicable to these other different cancer tissue types. The other thing to keep in mind is that my team and I, we've looked at other market segments as well. So I'll jump here. We also noticed that animal tissues also require lymph node dissection. So we have the veterinary markets available. And you can sort of see here that we have some of these other cancer tissue types that our device could be applicable to.
And moving to Alex, do you have a question?
Yes, first of all, again, I'll echo the comments that Lou made, that very nice presentation and a good solution to a real problem. One of the questions I have is whether or not you are planning in your IP strategy to look at somebody who might try to piggyback along your hardware sales and try to develop a competing software platform, especially since you're using a subscription model, where they might try to eat into your recurring revenue by providing an alternate software platform that can execute on the hardware.
Mm-hmm. Alex, that's a fantastic question. And I have to say that in terms of our IP strategy, we are definitely looking to work with one of the legal firms for IP counsel for sure. But there's a few things to keep in mind. Now, pathology is pretty unique from that standpoint, as you were mentioning. So there's a few factors that we have to consider. One is brand loyalty. So we know that with traditional, with a lot of the processes in pathology, a lot of the workflow is pretty traditional and manual. So this sort of presents a natural barrier for new entrants, because with us, what we're trying to do is we recognize this as a potential barrier. But for our go-to-market strategy, we're already partnering with teaching hospitals. And by working with them early on, we are working with the next generation pathology staff, and that we're hoping that would foster some sort of brand loyalty with our device. And that's sort of one of the aspects. We're also sort of incorporating the feedback from pathology staff into this as well. Now, maybe to answer your question in more detail, for other competitors, so if they were to replicate, let's say, the hardware or software aspect of our device, if they created a knockoff, there's an aspect known as vertical integration. And the thing to keep in mind is these competitor firms would have to have control over the supply and distribution of an open architecture USB ultrasound probe company. So to mitigate this barrier, we're actually working with an ultrasound OEM who is a market leader in open architecture USB ultrasound imaging. And they're the leading manufacturer of this ultrasound imaging solution. So for us, because we're working with them very closely, we're hoping to have an exclusive distribution arrangement established with this ultrasound OEM. So that would tie up a lot of the distribution channels for a lot of the competitors. And the few other things to keep in mind too is this network effect. So in pathology, it's a very close-knit community, and it's actually quite difficult for new competitors to enter the space without a critical mass of users. And especially this is more apparent when pathology staff may be more reluctant to use a competitor tool that their peers aren't using. So from the end of Tenomix, we're really hoping to leverage existing pathology networks within our team, engage the KOLs, and also present at key pathology conferences. I hope that answered your question.
Thank you.
Great, thank you. And I have some sort of very basic questions about the technology itself and what you're what you're actually finding. So when you go searching for the lymph nodes in this tissue, it's the reason for doing it is to help with staging the cancer, correct?
Sort of, Dr. Hamilton. So we're locating the lymph nodes in the tissues, but yes, you are correct that these lymph nodes are ultimately submitted for more tissue staining. So H&E staining, for example.
Right, right. So potentially you can be better than standard of care because you could find more. Okay, so here's my question. It's kind of philosophical almost. If you're better than standard of care, all the data collected to date is with standard of care. So does it turn up the whole staging on its end? Like, is your staging going to be different from the next hospital that does it the old way?
Dr. Hamilton, that's a fantastic question. And that's something that we're actively building on our study design. So right now, you're 100% right. We're in the sense that if they do miss finding the small lymph nodes, typically it's the lymph nodes between 1 to 5 millimeters that are difficult to find by hand. Theoretically, if they miss these lymph nodes, then the patient's cancer can't be staged accurately. So with our clinical studies that we're planning to develop with Hamilton Health Sciences, we would do a head-to-head comparison study with our device and in terms of the standard of care right now, which is its manual dissection.
Right, right. Have you engaged data scientists? Is there an AI element here as well to exploit?
Dr. Hamilton, for sure, that's a great question. So the AI element, we're approaching the software from a rules-based image processing detection method. So basically to better clarify what I mean by this is that we have sort of established certain set of rules for identifying these lymph nodes. So we know exactly what our algorithm is finding out. Down the road, a more AI or machine learned method could definitely be investigated. But from the standpoint of regulatory, it would be something that a rules-based detection method would provide more clarity on how our algorithm is actually picking up these lymph nodes.
Okay, great. We're almost out of time. Are there any other burning questions, Lou or Alex?
No, it's great. Thanks very much for the presentation.